So let's start with the parental generation. All homozygous for the marker loci. So these would be the Louisii flowers, homozygous for the markers. M is for marker, L is for Louisii. All of the cardinalis parents that are homozygous for the marker, so M is for marker, C for cardinalis, and also homozygous for the QTLs, so Q sub L is a QTL for the Louisii, homozygous for the cardinalis, QTL in the case of the cardinalis parents. So just to remind you, M is the marker alleles. These are the ones that we can genotype. These are the knowns. Q are the QTLs. These are the ones that actually cause the phenotype, but we do not know the genotype. We don't know the location. We don't know anything about them aside from we know that there are QTLs out there that cause differences in phenotype. So when we create our F1 generation, each individual is going to get a blue-black chromosome from one parent, a pink-yellow chromosome from another parent. If the marker is in linkage disequilibrium with the QTL, this is the scenario that we expect. All the F1s are going to be heterozygotes. When we get to the F2 generation, if the marker is in linkage disequilibrium with the QTL, there are only 4 different kinds of offspring that can be produced. You can get a blue-black from one parent and a blue-black from the other parent, and that represents this situation. You can get a blue-black from one parent and a pink-yellow from the other parent. That's the scenario in this box here. You can get a pink-yellow from this parent and a blue-black from this parent, this scenario here. Or you can get a pink-yellow from both parents down here. Because these are linked on the same chromosome, and because we're just talking about 2 generations, in our experimental situation with just 2 generations, the chances for recombination happening and causing these to get mixed up is minimal. And so we only have 2 of our 4 possible haplotypes, MLQL and MCQC. So this is the strongest possible linkage disequilibrium we could be in. This is only the result of the fact that we have all homozygote markers, all homozygote QTLs in both of these situations and then cross them. Now let's compare the marker genotype to the phenotype that we see. So if you think about this phenotype distribution, the phenotypes, remember, are caused by the QTLs. So if you have QL, your phenotype is going to be more toward this Louisii phenotype. If you have QC, the QTL that causes the cardinalis phenotype, you're going to be down on this end. So if you have the homozygous marker genotype, let's look at what that means for the QTL in this scenario. So these are the ones, this dark gray patch matching the dark gray distribution. If you're homozygous for the marker, you're automatically also homozygous for the QTL. So these blue ones are the marker genotypes, the black ones are the QTL genotypes. All of the marker homozygotes are also QTL homozygotes. You're going to get a phenotype that is the homozygous type phenotype. Why do you have a whole distribution? Because remember, this is just one QTL among many. The other QTLs are also having their influence in causing individuals to be sort of toward this end or toward this end, causing that normal distribution. Now let's look at the heterozygotes for the marker, represented by this medium gray. The heterozygotes at the marker are also all heterozygotes at the QTL locus. So that means that the phenotype is going to be intermediate between the Louisii and the cardinalis phenotype. It's going to be right in the middle. Now if we look at the homozygotes for the cardinalis marker, this light gray, all of the homozygotes for the cardinalis marker are also homozygotes for the cardinalis QTL, so we get a phenotype that tends to be more like the cardinalis phenotype. So in this case, because they're in linkage disequilibrium, we've got a strong association between the marker genotype and the phenotype position, whether it's more toward the Louisii or more toward the cardinalis phenotype. Let's look at what happens if the marker is not in linkage disequilibrium with the QTL. So in the case of our experimental situation, this would mean that they're not on the same chromosome. In real life, linkage disequilibrium can be stronger or less strong. It could mean that the marker and the QTL are on the same chromosome. but they're far enough away from each other that recombination has made them behave as if they're independently assorting. If the marker is not in linkage disequilibrium, if it's in linkage equilibrium or located on another chromosome, here's what we expect. Again, we've got the same code. These are the marker alleles. These are the QTL alleles. We cross the parentals to get the heterozygotes. So notice that these two aren't connected by the sticks the way they were in the linked situation. So now these heterozygotes are free to associate at random. They're going to undergo independent assortment. And so when we look at the F2 generation, they can combine in any possible combination. So these represent the different combinations of the paternal allele, all the possible combinations of the maternal alleles, and they can sort in any way. So we've got all four possible haplotypes represented as we would expect for a population that's in linkage equilibrium. And if we look at the relationship between the genotype at the marker and phenotype, let's first look at the homozygotes for the Lewisii marker. So the dark gray is represented by these dark gray boxes here. So you see that all of these are homozygous for this blue marker locus. And so the blue homozygotes can be associated with either the black Lewisii QTL homozygotes, heterozygotes on both sides, or the cardinalis homozygotes, which means that the full range of phenotype is possible associated with homozygous genotype at the marker. So this is represented by this full range of the phenotype. If we look at the heterozygotes at the marker, that's these medium gray boxes. So you see all of these are heterozygotes at this marker locus. Again, we see the same thing. Heterozygotes can be associated with homozygotes at the QTL of either kind, or heterozygotes. Again, individuals that are heterozygous at the marker can exhibit the full range of phenotype that's controlled by these QTLs because all combinations of the QTLs are represented. Finally, with the cardinalis homozygotes at the marker, these are these light gray boxes here, or homozygous pink. Again, we can be associated with homozygous black, homozygous yellow, or heterozygotes. We get the full range of phenotypic values in this case. So if we go back to our gas gauge analogy, this would be like you're trying to figure out whether you're going to run out of gas by looking at the speedometer. There's no relationship between the genotype at the marker locus and the genotype at the QTL. So the marker is not a reliable indicator of what is at the QTL. This indicates that they're not in linkage disequilibrium. That's not going to be associated with the QTL. So these researchers went through this whole process, identified all of the QTLs, figured out these relationships between markers and phenotype, were able to identify using candidate locus methods where the actual genes are for the QTLs and what those genes do. And these are the results they found. So let me just explain these graphs a little bit first. So on the x-axis, we have the percentage of variance explained. So that basically means how big an effect does that QTL have on the phenotype. And then on the y-axis, we've got the number of QTLs that explain that amount of the variation. So in answer to the question of whether we see many QTLs of small effect, that would be these ones over here, or a small number of QTLs with large effect, we see that the answer in this case is both. We've got a whole bunch of QTLs with small effect, and then in both cases, just a very small number of QTLs with large effect. So we need some more information to really be able to answer this question. So what the researchers did was they crossbred these plants in such a way that they got individuals that had all of the QTLs that are typical of Lewisii and just one of these QTLs with large effect from cardinalis, so that they could look at the effect of one trait at a time in an ecological context. So they bred these in the greenhouse and planted them out in nature and watched to see who came to pollinate the plants. So one allele that produced yellow pigment, Lewisii, this bee-pollinated flower, reduced the bee visits by 80% and also attracted a number of hummingbirds. So not as many as you would expect for something that's really fully adapted to attract hummingbirds, but they started to attract hummingbirds, and it very drastically decreased their attractiveness to bees. They found that one other allele that increased nectar production, so these hummingbird flowers tend to produce more nectar for larger visitors, and that actually doubled the hummingbird visits. These are very, very, very dramatic results. What this suggests is that a single mutation in a single individual could almost completely reverse the whole pattern of pollination. In flowers, when you are pollinated by different species, you're basically reproductively isolated. If a hummingbird is coming only to the kinds of flowers that hummingbirds like, then they're only going to cross plants that have those traits, right? So we've got a very small number of traits of very large effect that are producing almost immediate reproductive isolation. So if we think about your macroevolution topics, think about speciation, you have very, very rapid speciation in this case based on just a few small changes. So probably what we can say is that in the evolutionary history, the diversification of these flowers, it probably began with just one or two mutations of large effect that caused the pollinators to diverge, followed by a lot of tinkering, right? A lot of small adjustments that increased that effect and really made the cardinalis flowers fully attractive to hummingbirds in every possible way. So we've got a combination of the two, but the actual diversification in this case seems to have been caused by a small number of mutations of very large effect that caused a very rapid diversification.